Best greetings from Kyiv, the capital of Ukraine. Best greetings from Sofia, Bulgaria. Today we have stream between student and teacher. By the way, we have some kind of discussion about entomology. This is channel Dr. Victor Fursov, entomologist, beekeeper, teacher. And to have introduced myself, I say I'm entomologist. I'm research entomologist at the Institute of Zoology, National Academy of Sciences in Kyiv, Ukraine. I'm studying parasitic wasps of family Trichogramma and others belonging to Calcid wasp superfamily, Calcidoidea. And I have invited today for our stream student from Sofia, Bulgaria. He's still young, but he has advantage because he has a free time for some observations, free time for some careful collecting, free time for some very careful experiments with some insects. And he is devoted to mostly to biology and morphology, to study of beetles. I'm studying parasitoid wasps, and he's watching and observing, studying beetles. Bogdan from Bulgaria. So Bogdan, tell a few words about yourself, about your interest, and we can exchange our questions for today. Today we are planning to talk about parasitoids of beetles and moths. Tell a little bit about yourself.
Hello, everyone. I am Bogdan from Sofia, Bulgaria, and I am a young research entomologist who is studying the two orders of insects. The first order I started with was Orthoptera or crickets and grasshoppers, and the second one was the beetles, order Coleoptera, which I've been very impressed by because of the enormous different types of beetles, and the biodiversity of this group is just enormous, and it impressed me very much. So I choose to study the exact groups. I'm specializing in ground beetles, family Carabidae, and Dermestidae, the skin or dermestid, which are very interesting to me. And with dermestid beetles, I study Anthrenops, the carpet beetles. I know almost every species from this genus, and this year I'm going to breed only one species, but in the next few years I'm going to breed a bunch of new species that are not very well studied and not well known. So today I want to talk about some parasitoids of dermestid beetles and ladybugs. There are different types of parasitoids. I think almost all insect orders have parasitoids, but I specialize in beetles and grasshoppers. About grasshopper parasitoids, and I cannot say much. I know only the mites that attack different types of grasshoppers and locusts, but I know some parasites that are attacking different kinds of beetles. And I think mostly the first parasitoids I studied are those who are attacking ladybugs, the family Coccinellidae. So I'm going to talk more about them in this stream.
You plan to ask me something about parasitoids. Tell your questions,
Yes.
because you asked. I'm interested in parasitoids. I said, what kind of parasitoids? What groups? What is interesting? Because parasitoids, a lot of them. You see here behind of me on poster many parasitoids, and we have a different biology, different morphology. Tell your questions, and I will try to ask something
My first, yes.
to answer. Okay.
Okay, so my first question is about the genus Trichogramma. How many species of Trichogramma do exist? Is my first question.
What do you mean do exist?
I think, how many species are in the genus Trichogramma?
Okay. Let's talk about, in general, in parasitic Hymenoptera. Parasitic Hymenoptera, these are parasitoids of different families and superfamilies, by the way. Because, just a minute. Because initially we should say about insects. Some insects are phytophagous, by feeding on plants. Some insects are detritophagous, by feeding on dead plants or dead animals. And some insects can be considered as entomophagous, because they are feeding on other insects. So Trichogramma is entomophagous insect. There are different orders of parasitic insects, actually, by the way, not only Hymenoptera. There are some other orders which are parasitic. These can be Coleoptera, beetles, flies, Diptera, Neuroptera, Trichoptera, Psiptera, even some parasitic moths exist. Who are small parasitic Hymenoptera, micro-Hymenoptera? They are belonging to different superfamilies. These are Proctotrupoidea family, Beetlelidea family, Trigonelidea family, Cerafranoidea, Vespoidea, Apoidea, Cnipoidea, Evaneidea, Ichneumoniidea, Beetyloidea, Rusoidea, and Chalcidoidea. We should differentiate wasps as parasitoids. We should say that they are parasitoids because they are different from parasites. Parasite, this is organism which larvae usually developing on other organisms, but doesn't cause the death of host. And it can develop on different individuals of host. And it even may have some additional hosts. But parasitoids, this is special arthropod organism which are developing on host and kill the host, cause the death of host. So Trichogramma is causing the death of eggs. So this Trichogramma is egg parasitoids. But there are also some larval parasitoids, pupal parasitoids, even adult parasitoids. Even parasitism can be very different. Parasitism can be primary parasitism, just on first host. Then can be second parasitoid, secondary parasitism, and even next one, tertiary parasitism. Parasitism is a very large term because it can be also solitary parasitism and gregory parasitism. Solitary, when we, for example, in Trichogramma, in one egg, developing only one individual, and gregarious parasitism, when in one egg, in one host, developing many parasitoids. And Trichogramma is exclusively egg parasitoids because they are attacking insects of 11 orders from different orders of insects. But other parasitoids, like on this poster, they are different from superfamily Chalcidowasps. They can be egg parasitoids exclusively. They can be egg larval parasitoids. When parasitoids lay egg inside egg of host, and larva of parasitoid will develop inside larva. So it can continuously develop in parasitism. So not immediately, because egg is small and larva is big. So parasitoid laying eggs in very small egg, larva is growing, and inside larva developing many parasitoids. It will be, again, gregarious parasitism, or even some parthenogenetic polyembryonism, when an egg will multiplicate, and from one egg of inserted wasp can develop many parasitoids inside caterpillar or inside larva of beetle. So it can be also pupal parasitism or larval parasitism, and also can be egg pupal parasitism. Again, egg laid inside egg of host, and parasitoid will develop inside pupa. And regarding some coccinellid, we know about adult parasitism, when parasitoid attacking beetle, just the adult. Parasitism can be very different, especially parasitism can be internal parasitism, and it can be external parasitism. Internal parasitism is when larvae develop inside the host. Parasitoid lay eggs inside the host. For example, for Trichogramma laying egg inside the egg. But it can be also exoparasitism, external, when parasitoid lay egg on the surface of host. For example, Eolophid family Eolophidae, genus Entodon or genus Euderus, lay eggs on the surface of a caterpillar, and larvae are growing slowly on the body of caterpillar. So this is exoparasitism. And there are many examples of parasitism. Trichogramma parasitizing exclusively eggs of different insects belonging to 6 families, six orders, not families. For example, for family Trichogrammatidae, 11 orders. But Trichogramma can parasitize 6 orders. But primarily, first of all, Trichogramma attacks beetles, eggs, and butterflies and moths eggs. Mostly butterflies and moths. More than 20 families of different moths can be attacked by Trichogramma. So very wide trophic relations. Not only on one host, sometimes, especially in laboratory. In laboratory, if you can give with no choice experiment as a food, eggs of butterflies for Trichogramma will attack. Outside the laboratory, Trichogramma can select one family or other family. So there are some trophical relations. If Trichogramma-like family Noctuidae, she will go to bollworms, Noctuidae family. If she like tiny eggs of Papilionidae, she will go to Papilionidae family. But in laboratory, 200 species of caterpillar or butterflies and moths can be attacked by Trichogramma. Okay, next question.
My next question is about one species of Trichogramma, the most well-studied species of Trichogramma. What are the biology and ecology of Trichogramma evanescens? This is a very widely distributed species, and it's often sold in different markets about gardening and different stores for biological controlling agents.
There are some laboratories which are breeding Trichogramma on eggs of grain moths. Citatroga cerearella, this is a tiny moth which are bred in laboratory. These tiny moths, they produce eggs in mass number, in mass quantity, because this is laboratory. In laboratory, Trichogramma introduced to eggs, and she's parasitizing these eggs. So eggs before hatching Trichogramma are sold to farmers. So they're sold in different ways. For example, sometimes eggs of Citatroga glued on paper, and so it's sold just like special cards. And these cards can be distributed somewhere in farm field, in orchard or in farm field. Sometimes eggs attached to special tiny small capsules, glued inside, and capsules about this size capsules, carton capsules or paper capsules. They're covered by paraffin to prevent moisture, and inside will be eggs of Citatroga and some holes can be done, should be done on these capsules. So these capsules can be distributed in a field, in a cornfield, by machinery, so by special machine, or just manually. So Trichogramma, after distribution in a field, hatch from eggs from inside this capsule and crawling outside and crawling on the surface of plant. For example, corn, Trichogramma evanescens, mostly used in a cornfield, again, corn moths, Astrinia nobilalis,
Yeah.
and others.
Yes, this is a very well distributed pest, and there are many shops here in Bulgaria that sell this species, because this is a very problematic species in our cornfields. And the Trichogramma biocontrol is something relatively new for Bulgaria, and we in the past we we've never used biocontrolling agent. This is something new for us, and this species is slowly distributing here in Bulgaria, and it can be very very easy to get. So
okay, I know that, for example, I received some material from Bulgaria, so that's interesting to hear that you know it about from internet, from publications. So this is open market.
But what are the closely related species to Trichogramma evanescens, and how we can distinguish the different species of Trichogramma? Are there any key differences in which we can distinguish the different species more easily?
Okay, good question. Just on computer. For some people, I can show some pictures on computer. Okay, any pictures on computer? Wait a minute. I cannot share it on telephone, but on computer I can share some pictures with Trichogramma. As I said, Trichogrammatide is in there. They have hosts in 11 orders, in different orders of insects. And Trichogramma attacking eggs. But to identify Trichogramma, we need to use structures of antennae, mesosoma, metasoma, and mostly structures of male genitalia. Male genitalia is a small structure, especially because Trichogramma is small.
Yes.
Trichogramma parasitoid is just 0.5 millimeter. This is not a beetle. This is very tiny wasp, 0.5 millimeter. And male genitalia, this is 1/4 part. You must 0.5 millimeter divide four times. So that's why
Yes.
it is 0.1 millimeter.
Yes, I've read some of your PDF files, and I found out that you have discovered some species of Trichogramma, like in the 90s. So can you tell me more about those species that you have been discovered? For example, I think you've discovered one species named Trichogramma yawaria. Am I correct?
Yes, yes. This species was discovered in Japan. So we bred this species from eggs of some butterflies on rice inside the rice fields, for example. So we collected in a rice field this species and bred in laboratory, and I investigated this culture to study morphological structures of male genitalia to make identification, because species can be mostly identified by male genitalia and by DNA. But DNA, it needs special investigation. And also this is just in process. Some species are known for DNA study. So, but 200 species of Trichogramma have been described already, based on morphological studies. Some species we can consider as cryptic species, very close species, morphologically, with very tiny differences between male genitalia. Some species, like people say Trichogramma brassica and Trichogramma evanescens, very different on DNA study, but morphologically they are practically the same. But to another species we described with colleagues from Holland, we described from Panama. For this species, I described them based on morphological study, and our Dutch specialists, they added also DNA study. So we had morphology description, we had DNA description, and we have biology description. We published in the Journal of Natural History Museum in London, and they also found specific biological features Trichogramma in Panama. These were parasitoids of Helicanine butterflies with transparent wings, very famous one. And they found that Trichogramma can move on the body of these butterflies from one place to another place. Because butterfly is a big one, more or less big, and Trichogramma is very small. So Trichogramma is traveling on the body of butterfly. So this process is called forazy, traveling on another insect. Like some mites can travel on beetles. So Trichogramma can travel
Yes.
on butterflies.
And but some Trichogramma species are flightless, especially the males, and key difference maybe, because my next question is about how to distinguish male and female species of Trichogramma. Is it different? Is it difficult, or is it easy in some species?
Females are very difficult to distinguish. Females, very difficult to distinguish. But Trichogramma has also some tricky biology. For example, some lines, some species. For example, if you found Trichogramma and breeding in laboratory on eggs, we can receive only females. And so we cannot identify what's going on. So we try to check what is the species. Practically difficult only to make DNA study. But if we don't have opportunity to make immediately DNA, we can feed them with some antibiotics. In some cases, it works, because Trichogramma inside may have Wolbachia microorganisms, Wolbachia. And Wolbachia will determine the sex. If we
yes, so
just wait. If we treat Trichogramma with sugar and honey, with some tetracycline, with antibiotics, we will kill microorganisms inside. So Trichogramma will be healthy, so-called healthy. And next females will produce males and females. So we can check and differentiate this one species. But there is another species we can treat with antibiotics. But Trichogramma has no Wolbachia. And Trichogramma will produce only females. Only in some very rare natural conditions, sometimes males. But this is a problem, because for females, very difficult to identify.
So in nature, they are most likely parthenogenetic. So they are reproducing without males.
No. Most of species, we have both species, males and females. Majority of species, we have males and females. But some can be parthenogenetic. some species, not all. Only a few species, maybe
Yes.
1/10 part. One part among 10.
So do they have a great biocontrolling potential? So in your streams, you told us about the biocontrolling potential of the genus Trichogramma. So is it a great biocontrolling agent for different pest species of moths and beetles?
At the present time, we can say that Trichogramma is reared in about more than 20 countries. So more than 20 countries are involved in biocontrol. So of course, most important countries can be, say, China, India, maybe Germany, Bulgaria as well, Slovakia, Czechia, France, and also USA and Brazil. So quite many biological laboratories are rearing Trichogramma and selling it to farmers for biocontrol, for organic agriculture, because organic agriculture is very important, especially in Europe and America as well.
Yes, because there are no pesticides and the plants stay healthy, and the pest species are controlled by a natural predator that is parasitizing their eggs. But do all Trichogramma species attacking eggs, or there are different types of parasitoism in this group of wasps?
Trichogramma can also attack beetles. Trichogramma attack beetles, for example, some tortoise beetles of group Cassidinae. Trichogramma can attack Cassidinae eggs on plants in nature, but they occur not very often. But if you can collect them on plants, Cassidinae, they lay eggs in small groups always. Sometimes one by one, sometimes 2 eggs, but also always eggs were attached to the plant like scales. Beetles of Cassida lay eggs like scales, like scale insect, covered with some color. If you take off cover, you can see 3 eggs, 2 eggs, and different species lay different shape of eggs. Cassidina is very widely distributed, not only in semi-tropical region, in our region, but very large distribution in tropics. Many different species of Cassida are living in tropics, so many Trichogramma are still unknown in tropics, for sure, which are parasitoids of beetles.
Yes, and maybe my last question is about let me think about what is about Trichogrammatidae. How many other genera, and how many species of wasp are in the entire family Trichogrammatidae, and what can be said about other genera, not only Trichogramma, but other?
Well, I can count you some hosts of Trichogrammatidae as well. Wait a minute for discussion. I don't miss something interesting. Regarding family Trichogrammatidae, we can consider that probably about 400 or 500 species of Trichogrammatidae in the world, or maybe more, because they're very small and weakly studied in some regions. Because their size 1 millimeter, this is the biggest. Ordinary size about 0.5 millimeters, or smallest of Trichogrammatidae 0.2 millimeters. Genus Megaphragma, parasitoids of thrips. There are about 40 genera of Trichogrammatidae, and they develop on different hosts, as I said. Hosts can be very different, 11 orders, including, as I said, moths, Lepidoptera, beetles, Coleoptera. Also, grasshoppers are hosts, Orthoptera. Hosts, dragonflies, Odonata. So they can swim under the water, some parasitoids. Tiny Megaphragma, they are parasitoids of eggs of thrips, Tizanoptera order. Trichogramma parasitize eggs of lacewings, Neuroptera. Some tiny parasitoids, Trichogramma, parasitoids of bark lices, Psocoptera order. Very interesting parasitoids reared from alder flies or Megaloptera, which are living near lakes, near streams. Many Trichogramma and other genera are parasitoids of flies, Diptera, and also some parasitoids of leafhoppers, quite many of them, and even some bugs under the bark of trees, inside stems, inside leaves. So order Hemiptera or Homoptera, say. So totally quite very large scale of hosts. So that's why quite interesting to search for different hosts of insects. And if you can find eggs, there are some possibilities to find egg parasitoids. And this is a great possibility to find new species, because
Yes,
biology weakly studied. But if you collect eggs, you can collect not only one individual from a group of eggs, like 5 eggs. You can receive 5 specimens for study, because in a trap, in some collecting methods, we can find 1, 2 individuals, one by one. But from eggs, we can breed many, even 100 specimens, just from one egg group. For example, of Lepidoptera can be bred many individuals, 20 specimens, 30 specimens, because many insects lay eggs in groups. So this is great opportunity. But this is difficult, because it needs to be careful eyes, to be very observative, to search for eggs on plants, on stems, on seeds, inside plants, under the leaves, or under the leaves, for example. Sometimes eggs just on the surface, sometimes inside the tissue. Needs to be very careful. Or in many cases, eggs concealed. They are not invisible, so they covered with leaves. So it needs to open leaves. And there are some eggs, like for some leaf roller beetles. Leaf roller beetles, they're making leaves like a bowl, like a cigarette. Atellabidae.
yes, Atellabidae. And
So, but
they are very well
cigarette. They're making a roll of leaves and lay egg
to
inside.
protect the eggs. Yes.
Egg inside. So this leaf just this egg cigarette on plant. But there are some egg parasitoids that are coming from another plants. We're searching precisely for eggs of these leaf roller beetles. So we attack these leaf roller beetles with very long ovipositor. An egg of leaf roller beetles quite big, about 1 millimeter. So this genus Paropea, quite a big one, about 1 millimeter, with long ovipositor near 1 millimeter. So it can penetrate the leaf and can find egg inside this leaf roll and attack it. So biology is very different and very interesting. For many species, still unknown. So this is a great subject for the study of biology and trophical relations between eggs of insects and parasitoids.
So these were my questions. So in the next part of the stream, I'm going to talk about the parasitoids that I've studied and I know them to parasitize several kinds of beetles
Yeah, so
that
okay,
are interesting to me.
okay, you studied some Dermistidae, you studied some Coccinellidae. Which parasitoids did you find?
So the first parasitoid I found was the braconid wasp of that parasitize on coccinellid beetles. Its name is Dinocampus coccinelle, and it's very well known from different species of ladybugs, but including Coccinella septempunctata, Hippodamia tredecimpunctata, and Coleomegilla maculata. So they basically turned the ladybug into a zombie guarding the wasp cocoon until the next generation of wasps hatches. So Coleomegilla maculata is the most frequently attacked species of ladybug, and Dinocampus coccinelle has been described from this exact species. But about 25% of Coleomegilla maculata recover after the cocoon and after the imagination of the adult wasps. And this the mature first starts with the mature female wasp. She will sometimes oviposit in the male into male specimen of the ladybug, but mostly on female ladybugs and or on larval instars. So the eggs are oviposited inside the ladybug's abdomen, and the larvae hatch after 5 to 7 days. And they have large mandibles, and they dig inside and eat inside of the living larvae. So when the larvae develops, and then the adult ladybug exits the cocoon, the larva still feeds on the new the imago's body, and then it pupates, and the ladybug stays attached completely without moving, and the the cocoon is placed just under the ladybug, and in about 6 to 9 days, the wasp emerges from the cocoon, and remarkably, 25% of ladybugs recover from this paralysis. But some of them eventually died. I've seen some ladybugs in the past who were actually in my breeding. I had one ladybug that was infected with this parasitoid wasp, and it eventually died, but the parasitoid was released into the wild. And the interesting fact is that Dinocampus coccinelle can be itself parasitized by Gelyssa agilis, a hyperparasitoid wasp that is known to mimic ants and host
Wingless.
the cocoon.
Wingless. Has no
Yes,
wing.
the wingless. Yes, the wingless species Gelyssa agilis usually takes about twice as long to emerge as Dinocampus coccinelle, and Dinocampus coccinelle is actually not good because the average adult Aphidophagous ladybug can consume about 5,500 aphids in their life, and over 200 aphids in a day. So any ladybug parasite is bad for them, because they ladybugs are very beneficial to agriculture and to the gardens. the interesting fact was that Dinocampus coccinelle was described in 1802 by Schrank, who described this species as Ladybug Cure 2155. So they have small, slender body, green eyes, and front legs and the apex are petitionate with petitionate abdominal muscles. They sometimes Dinocampus coccinelle can attack the eggs of different kinds of ladybugs. The most frequently attacked species, as I said, Columbigium maculata, Hippodamia tredatim punctata, and Coccinella septem punctata. And
I have a question. I have a question. You said that Dinocampus larva is feeding on body of ladybug, and sometimes
yes,
ladybug is surviving. So you observe
yes.
the survival of ladybug. But did you observe? Does ladybug lay eggs after parasitism, or no eggs after parasitism?
No, actually, they attack. I think the old ones, the old ladybugs that are no longer reproducing. But sometimes they can attack larvae, and but these do not affect the future development of the larvae. Continues feeding on aphids, and then it spins a cocoon, and the larvae of Dinocampus coccinelle still lives and feeds on this cocoon, but the ladybug eventually emerges, and after, as I said, about 6 or 9 days, the larvae turns into a cocoon, but after it exits from the cocci of the ladybugs, they spin this cocoon, and the ladybug still it stays attached to guard this cocoon, and it's not moving, so it's staying. It stays, and it stays hungry, so it cannot eat, and eventually, if it stays for too long, it eventually dies. The interesting fact is that Dinocampus coccinelle causes this paralytic effect because it carries a RNA virus, Dinocampus coccinelle paralysis virus. So this virus is the cause of the paralysis of ladybug.
Paralysis, so making
Paralysis.
ladybug a zombie,
Yes.
kind of zombie. And also, I guess that after parasitism, old ladybugs, they do not lay eggs and they do not reproduce because they have huge damage. Do not feed and do not reproduce.
Yes. So
tell more about next group.
The next group, this is about domestic beetle parasitoid wasp. We are talking about the wasp from the family Beetlelidae. This wasp is called Lelius pedatus and is among the natural predators of Anthrenops verbas, and is well-studied gregarious parasitoid wasp. Upon discovering an Anthrenops verbasi larvae, a female wasp will land on the larva's dorsal side and attempt to line up its long ovipositor for paralyzing the larvae. So when it paralyzes, it lays its eggs under the larvae, but in this process, the larvae erect with the long hairs that they have on their abdomen and attempt to brush these hairs against the wasp. And the hairs detach on the from the larvae and attach to the wasp, and it gives the wasp a bit itching sensation. So sometimes the wasp can be out outcompeted, and the larvae can defend itself against the wasp. But eventually, if the wasp successfully attacks the larvae, it the larvae of the parasitic wasp lives inside the Anthrax verbaceae larva, and it attacks them. So by comparison, the closely related beetle species, Antrenus flavipes, which are with the high, slightly longer hairs, they are more protected in comparison from the Antrenus verbaceae, and the the larvae paralyze themselves after about 40 seconds. And the larvae of Antrenus verbaceae usually carries about 2 to 4 eggs of Lelius pedatus, and the eggs hatch in 3 to 4 days, and the larvae feed for 3 to 7 days, and actually they kill the host. So this is one of the controlling methods that are not very well studied. So the prevention and the prevention of infestation of the museum beetles actually can be done with this type of natural hosts without using any deep freezing methods and hermetically closing the drawers of insect collections and the windows, so we can use this species of wasp as a whole, as a biocontrolling agent
But
for.
question, did you collect yourself this
No,
parasitoid?
I didn't collect, but this species is known to be a parasitoid of different species, around 6 species of dermestid beetles, like Dermestis verbaceae, Dermestis zarnicus, Dermestis flavipes, and 3 species of Trogoderma. And it can be very effective.
Because sometimes I collect this Beetle in laboratory because we have collections and some insects. When we have cultures of different insects like Lariophagus and also some Beetle and Lariophagus pteramali developing on some beetles like Sterobium paniceum, like tiny drugstore beetle. So on drugstore beetles also can be developing this Beetle, tiny Beetle wasps. And sometimes, pretty often, it's in summertime, I can observe very tiny wasp Beetle crawling on my table, so I catch it. And sometimes when I come into the window, in my laboratory, and even in my house, at home, in my kitchen. So because we're crawling on the window, and we're tiny, about 5 millimeters size, very narrow and very flattened. Very flat, Beetle was very flattened. So we can go to some shelters of beetles for parasitization to find larvae. And sometimes if we can find larvae of beetle, we can catch larvae of beetle and crawl it like ant. We catch larvae of beetle, take it out of a concealed place, and then lay egg. Or sometimes we can catch larva, bring it, and bite it quite often. But this is very common behavior for many parasitoids. Biting the host. Beetle can bite the host and feed on the host by hemolymph, feeding on the hemolymph. Like Trichogramma is feeding, sometimes can make penetration with ovipositor and do not lay egg, just feeding on hemolymph. And beetle wasp can also bite the host, the larva of beetle, and feed on gamma just for egg development, for reproduction. So you can try to catch these tiny wasps in small tubes and then put them to the culture of your beetles.
And to see what's going to see the the parasitism in action, and
behavior, how they act, how they are doing parasitization, how they are moving, how they are touching by antenna, how they checking. This is my host. This is not. This is delicious host. Okay, and
yes,
this is not delicious. Okay, I will attack delicious one. Just lay eggs
yes,
in this one. So this one is good, and this one is small one, not good. I can bite it and can eat it little bit. So how do they make selection? So this large area for experiments in laboratory.
yes. The third parasitoid that I found is also a ladybug parasitoid. This is a genus of wasps, and I'm talking about one species of Phalacrotophora, a fly from the family Phoridae. I'm talking about Phalacrotophora fasciata. It's a parasitoid on ladybug larvae, especially prepupal larvae. It's flying near the larvae, and when it's near the larvae, it just lays one egg on the underside of the larvae, and when the egg hatches, the larvae burrow under the ladybug larvae and start eating it. And this species of fly mostly don't kill the host. So the host continues his life not very injured. So this is not very dangerous parasitoid. And another species of fly from the family Tachinidae, its name is Medina separata, is a parasitoid on adult ladybugs, and it lays one egg. The female lands on the adult ladybug, and then it lays one egg just in the middle between the pronotum and the abdomen of the ladybug, just in the middle of ladybug, in the underside, sometimes under the elytra, and then the larvae live inside this ladybug, and then the larvae continues its development, and then it spins a cocoon, and the adult fly just emerges from the ladybug. But this species kills its host, so it's dangerous for ladybugs. But there are not only different types of parasitoids, there are different types of fungi that are attacking ladybugs, and different predators like birds, dragonflies, spiders, and small predatory insects like grasshoppers and mantids.
Okay, I can say a few words that, for instance, you said also about some parasitoids and about your beetles. You said that some species of beetles you have studied maybe the first time, maybe because you said that somebody studied it, but very little information is known about some species of Anthrenops.
Yes, some species of Anthracnose are very, very poorly documented. Only their distribution is known to science, and my mission in the future will be to investigate more about them and to rear as many as I can, because I want to collect data for their life cycle, biology, and behavior. And Anthracnose is very important genus for me. It's very interesting because of the biology of the skin and nest feeding larvae, because as you know, the larvae are living inside the birds' nests, and the adult beetles have very interesting and beautiful colors of the scales on their elytra and their pronotum.
But I need to add a little bit that if you said that biology was not studied, so it depends from literature. So not always you can find full set of literature in internet. So because some species were studied maybe many years ago, maybe 100 years ago, 200 years ago, and some literature is very rare, published in some rare books which are not have not been scanned and have not been studied later. So that's why you must be careful to check information about some species in different resources.
Yes,
Because
is
some
it
books are very
really
difficult to find.
viable? Yes, most of the Anthracnose species are studied between 2000 and 2004 by a Czech taxonomist that was working very hard and was his best genus, and he described many species. I think over 50 or 60 species of Anthracnose are discovered by this Czech taxonomist.
Did he study Trichogramma species?
Yes, he studied Trogoderma, and also he studied some Dermestes species.
What is his name, if you?
I know his name by Hava. Its name is Hava. Yes.
And also, I would say about parasitoids. Not all information about parasitoids are available on internet and in Wikipedia, because parasitoids described more than 200 years ago some species, even by Linnaeus. But biology was very weakly studied maybe 100 years ago. And there are some special books for parasitoids and for predators. For example, there is a special set, about 7 books or even more, which are called a catalog of parasites and predators of terrestrial arthropods, host and prey and enemy, for Lepidoptera, for Coleoptera, for all several orders of insects. So this is very big catalog of parasites. For Europe, a special catalog, separate one. For North America. For Europe, it was counted by Herting and Simmons in catalog of parasites and predators. And it's quite interesting. You can search for name of host, for example, beetles, and you can find a list of parasitoids from different orders. Or host Lepidoptera, Macrolepidoptera, list species name of butterfly, and list of parasitoids. So this is like a very big catalog, very useful, but available not free on internet, only in some libraries, and some big zoological and biological libraries, or in some universities as well.
Yes, sometimes it's very hard to find information about the species you want to study, and I think the one of the best ways is to study yourself as more as you can. So I'm going to study some species of Anthracnose in the future. One of them is only found in Bulgaria and Greece, and it's very rare, and it's only found on around Black Sea and in the southern parts of the country. So I plan to visit these places and find these species and to collect some cultures of them and to breed them on the standard substrate and standard food material. But sometimes it's very hard to obtain breeding material of antennas because males and females can only be distinguished by the size and the form of the antennae. The antennae of males are small, and sometimes they have a bigger club on the bigger end of the antennae, and the females have more slim, a bit more thin antennae. So these are my plans about antennas. So there are many parasites and parasitoids, maybe on different kinds of trips. You told me about the genus of Trichogramma that attacks trips, the order Tisanoptera. So
Trips eggs.
trips, yes.
They're very tiny, 0.2 millimeters. This is a trips. An egg of trips is like this, 0.2 millimeter. Trips just 0.6 millimeter size. An egg 0.2. And parasitoid is very tiny. But it's possible to find either to rear from trips or collect only inside yellow pen traps. But from trips, needs to be careful because trips lay eggs. Some trips lay eggs on the surface of plants, but some trips lay eggs inside the leaves, inside where concealed eggs. So they're drying very easily. So it needs to collect piece of leaf, put it inside some cages with to keep some moisture, and just wait. So some parasitoids can emerge from eggs. Bulgaria has a more or less warm climate. Some parasitoids were found in Israel and other Middle Asia countries. So possibly megaphragma exists in Bulgaria as well. It can be interesting discovery for Bulgaria. Egg parasitoids of trips and larval parasitoids as well. Ceranizus eulophidae attack larvae of trips. So they're also interesting for study.
Yes, it can be very interesting. More people have to study parasites and parasitoids. I think I have one question for you. Do you have plans for traveling to warmer countries and to study their local parasites of Trichogramma or Beetle or the other superfamilies and families of parasitoid wasps?
That's a very good question, by the way, because for entomologists, for biologists, traveling and collecting is very important. But now in Ukraine we have a hard time with war between Russia and Ukraine, and situation is very complicated. Very hard time for Ukraine. So it depends from situation and depends from funds, because it needs to find financial support for this study. But I have been traveling to some countries. I have collected some material, and even in winter time we are studying our collection. And in summertime, entomologists usually collecting material. This is hard time for collecting material. So everything is possible. It will be a great idea to visit some countries to collect egg parasitoids and other parasitoids, because when you are traveling to another country where some groups are not studied at all, so you can feel yourself as Alexander von Humboldt from Germany, how he came to South America and he was the first discoverer of hundreds species of animals, big animals, small animals, tiny animals, plants.
Yes.
Everything
And
was new.
uh,
So that's why I wish you, if you can find opportunity in future to visit some tropical countries, you can find big discoveries and small discoveries as well. So let's think about it. Everything is possible. Let's hope for new entomological expeditions.
yes, it would be a great idea.
Thank you for our meeting. If some people are coming here for the first time and may have some questions, you can contact me on my email. So this email is also indicated under the description of this video for some questions about insects in your garden, in your orchard, or some questions about parasitoids, which you can find, which you can collect in your country. Because in some countries, parasitoids are very weakly studied because they are very small. If you have some questions about Bogdan's study about his beetles, I give a link for his channel under the description of video. You can go to his channel and check his work, his observations, his presentations on his YouTube channel. So welcome to visit our channels. Mine in Ukraine and his channel in Bulgaria. Thank you for your attention. See you soon on my channel. Thank you very much, Bogdan, for coming. Thank you for presentation. Thank you for discussion.
Thank you too. I'm happy to be here. Every time you make streams like this, I'm happy to be here and to talk to you about different themes and different things about insects. So it would be great to talk again in the future. So see you soon in the next streams.
Thank you very much. My great pleasure to hear that I gave you a little bit enthusiasm and motivation. Motivation to study your insects, motivation to study literature, and motivation to learn more about insects. Thank you for your enthusiasm and curiosity and interest in biology and entomology. Hopefully, some other students can see you as example of young entomologists and can join
Yes.
the team of entomologists as well. So,
So
welcome to
it would be
study
great.
entomology. Okay, see you soon in next streams. Bye-bye.
See you soon.
Have a
Have a
great day
great
too.
day.